Hello dear learners, welcome to my channel Embrace Biota. In today's video, we are covering the important points related to the cell biology. Firstly, we will start with the discovery of cell, the contribution of different cell biologists, the structure of the cell wall, cell membranes, and cell organelles. First topic of discussion is discovery of cell. First, the discovery of dead cell occurred. The dead cell was discovered by Robert Hooke in 1665. He observed the cork cell of Indian oak known as Quercus sober and identified a honeycomb-like structure which looks like the little room. He wrote about his work in his book Micrographia. With advancement of the microscopy technique, the living cell was discovered by Anton von Leeuwenhoek. He took the pond water and saw many rod-like bacteria in the water. These were the bacillus bacteria and he named them as wild animalcule. The book published by Leeuwenhoek is The Secret of Nature. After that, the discovery of nucleus was done by Robert Brown. The cell theory is proposed by the German botanist Sleeden and British geologist Swan. Sleeden studied only plant cell and Swan studied both plant and animal cell. According to Sleeden, the body of plant is made up of cell. Swan differentiated plant and animal cell on the basis of their outer boundary. In plants, the outer boundary is of cell wall and in animals, it is of cell membrane. They combine their result and give the postulates of cell theory. First one is the cell is structural and functional unit of life. Next is the all the living organisms is made up of cell. Only exception to the cell theory is viruses, as they are acellular. The cell theory was criticized because they failed to show that how new cells are formed. The answer to this question is modern cell theory, also known as cell lineage theory or final shape of cell theory. Modern cell theory was proposed by Rudolf Virchow. He gave the term omnicellula e cellula, which means the new cells arise from the pre-existing cells. And this is the final shape of our cell theory. There were many cell biologists related with the important discoveries of the cellular structures. The Robert Brown is credited with discovery of cell nucleus. Purkinje gave the concept of protoplasm. Hofmeister is known for chromosomal discovery. Sleeden and Swan formulated the cell theory. Robert Virchow gave the term omnicellula e cellula. Walter Fleming gave the description about chromatin, and the Christian Gram performed the Gram staining. Now we are going to study about cell wall. The cell wall is the outer boundary of the cell. Exception lies in the animal cell, where the cell wall is absent. In the bacteria, this cell wall is made up of peptidoglycan. The peptidoglycan is polymer of N-acetyl glucosamine, that is NAG, and N-acetyl muramic acid, that is NAM. So it is also known as murine. In algae, mostly the cellulosic cell wall is seen. But in diatoms, this cell wall is silicaceous, and in Chara, there is deposition of calcium carbonate in cell wall. The fungus cell wall is made up of chitin, which is also a polymer of NAG. In higher plants, the cellulosic cell wall is seen. Talking about the composition of plant cell wall, it is rich in cellulose. Also, hemicellulose, pectin, lignin, suberin and cutin is there. Cell wall consists of the matrix and cellulose fibrils. This diagram shows the structure of cell wall. The primary cell wall is the outermost wall layer. Inside to it is the secondary cell wall. The secondary cell wall is thickest among all. This secondary cell wall is further divided into three regions, out of which the middle region, which is S2, is the most thickest. Inside secondary cell wall, in some cases, the tertiary cell wall is also seen. The two plant cells join to each other with the help of middle lamella, and the cells communicate with each other with the help of plasmodesmata. Plasmodesmata connects the cytoplasm of two different cells. Inside the plasmodesmata, there is desmotavule. This desmotavule is the part of endoplasmic reticulum. It is only present in the plant cell and absent in the animal cell. So the gap junction which connects the plant cell is known as plasmodesmata, whereas in case of animals it is called as desmosomes. So the cell wall of plants is divided into different wall layers. First wall layer is the primary cell wall composed of cellulose, hemicellulose and pectin. The primary cell wall is living in nature. Inner to the primary cell wall, the secondary cell wall layer is formed. The secondary cell wall is basically the mature primary cell wall. Secondary wall layer is the thickest layer of the cell wall. Thickening is due to the presence of lignin and suberin. Due to the lignification, the secondary cell wall is dead. Inner to the secondary cell wall, the tertiary wall layer is formed. It is made up of the polymer of xylose or xylanes. It is seen in the tracheids and in some gymnosperms. The partition between two cells is the middle lamella. Middle lamella is composed of calcium pectate or magnesium pectate. Now we will see how the growth occurs in the cell wall. Two types of growths are seen. First is the intersusception. Second is apposition. In intersusception, the addition of new cell wall material occurs inside the existing cell wall. This growth inside the existing cell wall occurs in the form of patches. It is feature of both primary as well as secondary cell wall. Next is the apposition. The addition of new wall material occurs outside the existing cell wall. This addition occurs in the form of layers. It is the feature of only secondary cell wall. Our next topic is cell membrane, also known as plasma membrane. The different terms are assigned by the different scientists for the plasma membrane. The term plasma membrane was given by Pfeffer. It is called as cell membrane by Nageli and Kramer. Robertson gave the term unit membrane for the plasma membrane. Plough called it as plasma lemma. So when we talk about plasma membrane, we can use all these terminologies. Various models of plasma membrane are proposed by various scientists. First one is the lipid model of plasma membrane. According to the lipid models, the plasma membrane is only composed of the lipid. These lipids can be present in monolayer, that is single layer, or bilayered. The lipid monolayer model is given by Overton. But according to the Grotter and Grendel, the lipid is present in the form of bilayer. After the lipid model of plasma membrane, the sandwich model was proposed by Daniel and Davison. According to them, the plasma membrane is made up of lipid as well as proteins. There is outer layer of protein and inside the protein lipid bilayer is present. So the lipid appear as they are sandwiched between the protein layers. The third model is the unit membrane model given by Robertson. He called plasma membrane as the unit membrane. He called plasma membrane as tripartile, means having three parts. The outer two parts are protein and inner is the lipid. The diameter of the protein part is 20 angstroms and of lipid is 35 angstrom. And the total diameter of plasma membrane is 75 angstroms. Many times the question is asked that who assigned plasma membrane as unit membrane. Then we have the fluid mosaic model of plasma membrane. It is the most accepted model of plasma membrane, proposed by Singer and Nicholson. They studied the plasma membrane of RBC. In the RBC plasma membrane, the lipid content is 40%, protein 58% and carbohydrate is 2%. They compared the plasma membrane with the glaciers floating into the ocean. And this ocean is of lipid and the glacier in the ocean are proteins. According to them, the plasma membrane has quasi-fluid nature. It means that the plasma membrane is dynamic and there is the free movement of the proteins. The quasi-fluid nature of the plasma membrane is attributed by lipids. Membrane has three components, that is lipid bilayer, proteins and carbohydrates. This diagram shows the structure of plasma membrane. Here the lipids are arranged in the form of bilayer and protein is incorporated between the lipids. The carbohydrate is also present, but it is towards the extracellular fluid. The carbohydrate present in the form of oligosaccharides, that is always conjugated. It may be either attached to the protein or lipid. Carbohydrate attached with the protein is known as glycoprotein, and when attached to the lipid is known as glycolipid. The protein may present on the extracellular fluid on the external side or may be intrinsic. The protein which is present toward the extracellular fluid is known as extrinsic or peripheral protein. The protein which is inside the intracellular fluid or incorporated in the lipid is known as intrinsic protein. If the protein crosses the entire lipid layer, it is known as transmembrane protein. The protein present is the alpha helical protein. The most abundant lipid in the plasma membrane is the phospholipid. Out of phospholipid, the major one is lecithin. The lipid is amphipathic in nature. It is composed of hydrophobic tail and hydrophilic head. The hydrophilic head is toward the extracellular fluid and the hydrophobic tail is toward the intercellular side. Another important thing is that the plasma membrane of inner mitochondria membrane is composed of the cardiolipid, whereas the outer mitochondrial membrane as well as the plastid and the gram-negative bacterial cell wall is composed of porine protein. This protein creates the pore in the plasma membrane and hence these membranes are the more permeable than the inner membranes. Next is the movement of molecule in the plasma membrane. Two types of movement occur. First one is the lateral movement. Second is transitional or flip-flop movement. In the lateral movement, the molecule moves within the same layer. It can be shown by protein as well as lipid. Lateral movement can be detected by using Frapp technique. Whereas in transitional movement, as the other name suggests the flip-flop, there is movement of molecule across the layer. Most important thing is that this movement is only shown by lipid and the proteins never flip-flop. Next concept is the protoplasm, also known as cytoplasm. Presence of protoplasm was first observed by Cotai. Dujardin called it as sarcode. And the term protoplasm is composed by Purkinje. Huxley called the protoplasm as physical basis of life. Protoplasm is the main arena where the cellular activities occurs. Its pH is 6.8, so is slightly acidic. The most abundant molecule present in the protoplasm is the oxygen. And the most abundant compound is water, followed by protein, then nucleic acid, carbohydrate and lipids. Up next is composition of protoplasm. Protoplasm is rich in water and proteins, so appears the colloidal. Many theories are proposed regarding the physical nature of protoplasm. According to Bilson, protoplasm is colloidal. Most accepted theory is the crystallocolloidal proposed by the Kolliker. Now we will discuss the cell organelles, that is small organs within the cell. Cell organelles on the basis of membrane is divided into two categories. First one is the non-membranous cell organelle and another one is the membrane-bounded cell organelles. Non-membranous cell organelle is ribosome, centrosome and nucleolus. Membranous cell organelles can be single membrane or double membrane. The single membranous organelles are endoplasmic reticulum, Golgi complex, lysosome, peroxisome, cytoskeleton, vacuole, glyoxysome and spherosome. Double membranous cell organelles are nucleus, mitochondria and plastids. The P or A in bracket indicate whether these organelles are present in plant cells, animal cells or in both. In today's lecture, we will only discuss about the double membranous cell organelles, only mitochondria and plastid. Rest will be covered in the next lecture. Both the mitochondria and plastid are semi-autonomous. Means these have their own DNA, but also remain dependent on the nucleus for their proper functioning. First one is the mitochondria. We know that mitochondria is known as powerhouse of the cell due to the production of ATP. Mitochondria are also known as chondrosomes or chondromite. Only one mitochondria per cell is seen in the microstereous algae. The maximum mitochondria is present in the chaos. Here, the 5 lakh mitochondria is seen in one cell. Mitochondria was first observed by the Colliker. He observed it in the striated muscles of the leg of insect. He named mitochondria as sarcosome. Mitochondria is semi-autonomous cell organelle, so it has its own DNA. The DNA in mitochondria is single, circular and double-stranded. Also, it is without histone protein and known as mitochondrial DNA. Mitochondria without outer mitochondrial membrane is known as mitoplast. The staining of mitochondria is done with the genus B. green. The most important thing is that the marker enzyme of mitochondria is succinate dehydrogenase. It is present in inner mitochondrial membrane. Mitochondria in flagellates is known as kinetoplast and the mitochondrial DNA is called as kinetoplastic DNA. Smallest mitochondria is seen in the Plasmodium falciparum. The largest mitochondria are in the cardiac muscles because cardiac muscle has to do the work continuously. Another important thing is that the mitochondria is absent in the Trichomonas vaginalis. Instead of mitochondria, Hydrogenomonas is present. It pumps the protein for ATP formation. माइटोकॉन्ड्रिया में राइबोसोम्स प्लांट के केस में 70S है and in animals it is 55S. There are two layers of mitochondria, the outer mitochondrial membrane and inner mitochondrial membrane. Outer membrane is porous due to the presence of porine protein and in inner mitochondrial membrane the cardiolipid is seen. This diagram shows the structure of mitochondria. Two membranes are there. Mitochondria is double membranous organelle, outer mitochondrial membrane and inner mitochondrial membrane. Between these two membranes there is perimitochondrial space, also known as intermitochondrial space. Inner mitochondrial membrane is folded inward and forms the finger-like projection known as cristae. Inside inner mitochondrial membrane there is mitochondrial matrix. The oxysome known as F0 F1 particle is attached to the inner mitochondrial membrane and towards the matrix. Inside the matrix, the mitochondrial DNA and ribosome is seen. So the outer mitochondrial membrane is more permeable due to porine protein. Inner consists of the cardiolipid and the foldings of inner mitochondrial membrane is cristae. These folding contains the F0 F1 particles inward. The mitochondrial membrane also contains the electron transport chain. In the perimitochondrial space there is high proton concentration, due to which there is low pH. And mitochondrial matrix has high pH compared to the perimitochondrial space. It consists ribosome, circular DNA and RNAs are present. All the mitochondria of a cell is together termed as chondriosome. The term oxysome is given by Lehenger and discovery is done by the Fernandez-Moran. That is why known as FOF1 particle. This is also known as elementary particle. The FO is toward the membrane and the F1 is toward the peripheral site. Here the FO is responsible for the movement of the proton and forms the proton gradient. F1 deals with the synthesis of ATP. In FO, the O stands for the oligomycin. Oxysome is also known as smallest rotatory machine of entire universe. अगर माइटोकॉन्ड्रिया के फंक्शन की बात करें तो माइटोकॉन्ड्रिया इज द साइट ऑफ एरोबिक रेस्पिरेशन. Also ATP synthesis occur in the mitochondria. Mitochondria also provide site for the Krebs cycle. In plants, mitochondria is also site of photorespiration. The role of mitochondria in cellular respiration was first demonstrated by Michaelis and Kingsbury. Next organelle is plastid. Plastid is a double membranous cell organelle just like mitochondria. Term plastid is given by Haeckel and discovery is done by Skimper. Plastid is present only in plant cells and euglenoids. It is absent in animal cell. There are three categories of plastids are known. First one is the chloroplast, which is of green color. Second is chromoplast. The chromoplasts are different color due to the presence of pigments. And the leucoplast form of the plastid is colorless and meant for only storage. It is further divided into amyloplast, aleuroplast and elioplast. The chloroplast is known as the kitchen of plant cell. It contains the stalk of thylakoid and which arranges to form the grana. Chloroplast contains the pigment that is chlorophyll. Chlorophyll is lipid-soluble pigment. Various forms of the chlorophyll exist in different plant groups, but the chlorophyll A is universal, present in all. We will discuss more about chloroplast in plant physiology. Second type of plastid is chromoplast. Chromoplasts are of different color due to the presence of pigments. These are responsible for providing the color to the flowers and fruits. Some major examples of chromoplast are rhodoplast, present in red alga, pheoplast, seen in brown algae, and the most important is chromatophore, which is present in blue-green algae. Last category is leucoplast. These are the colorless plastid and meant for the storage only. Example is amyloplast, which stores the carbohydrates, aleuroplast for storage of proteins, and elioplast for the storage of oil and lipids. This is all about today's lecture. If you find this video helpful, subscribe the channel. My name is Anna Sharma and thanks for watching.